Okay, hi, I'm Gary Bader. I'm a professor of computational biology at the Donnelly Center, which is this building on the University of Toronto campus. This is a interdisciplinary research building with lots of different departments represented, and it focuses on fundamental biology research. So I'm going to talk today about an idea called mapping the multiscale human. And this is an idea that's coming out of a funding program that we have from a funding agency called CFAR. CFAR is the Canadian Institute for Advanced Research, and it's a unique type of funding agency that just funds convening people at workshops to over a long period of time to discuss particular ideas. It was some of the programs that CFAR has funded have lasted for decades. One of them that's lasted for 40 years is credited with inventing deep learning. So it's an interesting opportunity to discuss specific deeper topics for an extended period of time. So the topic that we're focusing on with all of these people listed here, you can go find out more about it at this URL, is the multiscale human. And the goal is to understand how the human body works across scales, from molecules to organs and all the way to the whole body. And ideally that would improve our understanding of diseases and ability to predict their risk and treatment. So traditionally, people who are trying to understand the human body have kind of been separated in different silos across scales. So the molecular and cell biology community has focused on understanding molecules and molecules inside the cell all the way up to a single cell, whereas physiology and most of medicine has focused on the whole body as a system and not even thought about cells, for instance. So, and then they don't really talk to each other very much, even though they're studying the same system. So a new technologies such as single cell and spatial genomics which provide a way of measuring lots of information about millions of cells, including their spatial positions in the body, has created an interesting opportunity to create a bridge via multicellular tissues between these fields. So one of the questions we were trying to ask is, can we develop a unified field that considers how the whole body works across scales? And one of the ideas that's come up so far is that maybe we can think about using generative AI, which underlies technology like ChatGPT, to understand the human body. And we can think about the genome as the ultimate generative model. And the kind of joke is that it's the ultimate model because the genome generates humans and humans create AI. But the more serious version of it is that maybe we can use generative AI to kind of take all of the measurements that we make for the human body, learn how to recreate systems in the body, and potentially have those generative AI systems learning actual mechanisms. You can kind of, an interesting kind of question that comes out of this is, if you have a generative AI system that takes a bunch of images of people and it learns to generate more people, just images of them, like people's faces, and you've seen this online. Is it actually learning something about how the human body is structured, or is it just copying and pasting little bits of images together? So I think that's a very interesting fundamental question. If it's just even if it's just thinking, learning how to copy bits and pieces together to make new images of people, is it can we figure out how to change the technology so that it actually learns something fundamental? So another thing that comes out of just thinking about this as the genome as the ultimate generative model is that if the genome plus the environment gives rise to the human body, then everything that we measure about the human body at every scale, whether it's single cell information, this little plot is a bunch of different cells and their relationships to each other based on their similar gene expression profiles, MRI images, or even all of the text that is written in publications about how the human body works in any scale, it all derives from the genome, from a common source of information, and thus should be correlated. And maybe we can study how these data layers are correlated with each other and create a unified model that bridges scales. Okay, so we can ask, there's a little bit of a conceptual exploration of this idea. We can ask, is machine learning a good approach for understanding the body? And I would argue yes, because the human body and all biological systems that we know about have evolved through two major operations, two fundamental operations, which is copying and mutating. So every cell is copied from predecessor cells. That provides a huge amount of redundancy in biological systems. So when we make measurements over and over again in biological systems, we're going to see the same things over and over again. And that's actually great for machine learning because redundancy helps with pattern recognition. It kind of reinforces the signal. And then mutations, which are also fundamental in evolution, create variation. So every human is different. There's variation in the population. And that variation is also critical because without variation, we wouldn't be able to do correlation or prediction of how, if you're thinking about regression, you have a set of measurements and you try to see if the set of measurements that you make predict some outcome. You would not be able to do that if you didn't have variation. So, I think machine learning is actually a pretty good match conceptually for thinking about biological systems. Machine learning traditionally is a black box, a lot of different types of machine learning. So we'll have to figure out how to gain mechanistic insight from using it in this way and also consider multiple scales. There is a limitation because rare events will probably require, we might not be able to model very rare events using this approach. It might still require some mechanism-based interpretation. Okay, and then another interesting aspect of existing data is genetics. And it's interesting because it maps information across scales, so it links scales together. So when we find that a mutation causes a disease, we can trace that effect of that mutation across all the scales. We know where the mutation is in the genome. We know that it affects a protein, a complex, a pathway, a cell, a tissue, all the way up to the whole body. And there's huge amounts of information available about human genetics in large biobanks. And we can try to, one of the challenges is to use this information in combination with machine learning and mechanistic understanding or mechanistic modeling to create a bigger system, a more holistic view of the system. So there are a lot of projects around that are mapping the human body. The Human Cell Atlas is one that's trying to map all of the cells in the human body, and there's other ones. And that kind of maps the structure of the system. So we know where all the different cell types and where they all live. One of the things that we need to think about as well is how we map the function of the system, even define the function of the system. And a generative model might help with this. It would need to be mechanistic. So as I mentioned before, we need to have, we need to understand how it works. We can't just let the AI understand and not let us understand. It would have to be predictive, accurately predictive, like be able to predict the response of the system to perturbation, be able to link scales, like predict, emergent properties that occur at a higher level of, a higher scale compared to a lower scale. And there are lots of interesting medical applications if we can get this to work, like digital twins, which is an idea that, you could take parameters that are measured from an individual and parameterize a model, computational model, and use that model to make predictions or even test drugs and other things without testing it on a real individual. It's a little bit sci-fi, but a lot of people are thinking about that idea. Okay, so I'm going to just go over a few different example projects that touch this idea in a few different places. So first of all, I'm going to talk about the nanoscale of the human body. And this is a project with a number of different people in the University of Toronto to try to understand the human liver at the nanoscale resolution. So the pixels of these 3D images are like 1 nanometer or 2 nanometers big. And the way that these images are taken is to use volumetric electron microscopy. So the idea is that you have a tissue block in a plastic resin, and you use an electron microscope to take a very high resolution image of the top of the block, and then you shave off a layer, and then you repeat, and you get this nice Z stack as a result of 2D images that you can put together into a 3D image. And this is the kind of thing that results. And the challenge is to kind of go from this grayscale block of pixel variation to actual cell boundaries and cell annotations. So now each of these are cells. And the way that that traditionally works is that manual tracing is done. And it takes thousands of hours per image. And so it's extremely difficult to do. Here's kind of an interesting kind of view of one of these 2D images. You can see all this amazing structure inside of a cell. And here's a kind of animation that's going through the Z stack of the images that we've taken of a human liver, or sorry, a liver sample at high resolution. So how do you do this annotation? How do we interpret these images if the annotation, if manual annotation takes thousands of hours per image? It's incredibly impractical. So ideally, we'd be able to automate it. And a lot of people are, a number of groups are working on using machine learning to automate the identification of all the different segments or objects in the image. And we worked on one that we call Maester, and I'll just explain how it works. This is done by a student a PhD student in my group, Ronald Shea. The way that this works is that we take these 2D images and we split them up into little patches, and then we hide some of the patches, and then we give it to a machine learning system that I'll explain in a sec, that tries to reconstruct the image. So the way that this, and try to replace the patches that were hidden. And then we can test, because we have the patch that we hid, we can test how good that system is at reconstructing the missing patch. The way that this works is that the images are fed to an encoder. This is an autoencoder system. So the way the autoencoders work is they have an encoder, a reduced dimensional model in the middle that is learned, and a decoder. So the encoder takes information that you have, in this case images, and encodes it as a vector. And then the vector starts out being just kind of introduced. We reduce the dimensionality of the vector to this internal layer, and then it gets decoded back from a vector to an image, and then you test whether the resulting image, how good it is at reconstructing the original. And you use that reconstruction loss function to iteratively train the autoencoder so that it eventually learns to do well at this task. And once it's learned that, the internal representation is meant to ideally capturing something about the essence of the problem. And then it can use that to reconstruct accurately these things. So once we've done that, we take the internal representation, this is a kind of 2D view of it, and we cluster it. And these different colors are meant to be different clusters. And then when we plot the colors of the clusters on the image, you can see that it actually identifies all sorts of interesting cellular structures. And you can kind of see how well it's working with this, how well the masked autoencoder is working. So if we have original images with mitochondria and different types of cellular structures, and we mask 25% of the image randomly, and we ask it to reconstruct, here's the reconstruction, here's the original image, and you can see that's pretty close. If we remove 50%, it's actually still get some of the image, and even up to 80% masking, there's like a blurry version of it, but you can see that's actually still capturing major features. So these autoencoders are pretty good at automatically learning how to reconstruct the original data. And this is a form of self-supervision or unsupervised machine learning, which is similar to how ChatGPT is trained. A big advantage of this type of machine learning is that you don't have to make manually create training data. You just give it a bunch of data and it automatically learns how to do its task, which is exactly how ChatGPT works. Which if you basically does something very similar, except it masks the next word in a sentence, and the model tries to learn how to predict the next word that you hid. And if you have billions of sentences, eventually it learns to be able to have a coherent conversation with humans. Here is kind of the original electron microscope image of a cell. This is just a test data set that's available in the community to develop methods on. This is the segmentation of this cell that was manually created, and this is what our method does. It's pretty good. There's a lot of fuzziness and it's not perfect always, so there's room for improvement here. The previous method that tried to do this looks like this, so we're way better than that, but definitely not completely solving the problem. So that is kind of an interesting way of kind of mapping very detailed information about the human body. And we were trying to automate this so we can do that much more high throughput and cover more samples. The second example project that I'm going to tell you about is again with Bo Wang, who's a machine learning researcher at the University of Toronto and the Vector Institute. Quan was a undergrad student, and Ronald is my PhD student. And they worked on this model for trying to predict the correlation between images and gene expression data. So I mentioned in the very beginning that if all the information in the human body, other than the parts that are kind of influenced by the environment, are encoded in your genome, that any measurement that we make should be correlated. So this is an example of a project where we're trying to automatically identify these correlations using machine learning. So the idea here is that we can take technology called spatial transcriptomics. Spatial transcriptomics measures a lot of information about a tissue slice. So we can take like a tissue slice here. This is a slice of a human liver. If you zoom in, you kind of see cells, but it's not very high resolution. In addition to this image, we get a whole bunch of transcriptomic measurements. Each of these dots basically represents a whole transcriptome that's measured for the set of cells that are under that dot. And usually it's, say, 2 to 10 cells per dot. So it's not totally single cell, but we get a lot of information about how the transcription of genes, like this gene GLUL or this one, varies over the liver tissue. And so what we wanted to do is try to predict one from the other. And we're wondering actually if we could take an image, try to predict these patterns just based on the image. And that would help us understand how the image and the gene expression data are related. So we used a method called, a type of machine learning method called the joint embedding method that was influenced from a method called CLIP, or contrastive loss image processing. So the idea here, so the joint embedding is the kind of general idea or general type of machine learning method. So the idea is that we take images and we embed them. Embedding them means creating a multidimensional representation. You choose the number of dimensions you want to represent the data in. Similar to the previous project, this embedding space, the space of relationships between all of the data points in the image is learned automatically. And we also take the expression profiles, these spatial expression profiles, and encode them as vectors, and then learn their embedding or latent space, which is just like the hidden space of relationships that could kind of characterize the original data. But we do this in a way where each embedding is aligned using something called a contrastive loss. So we have for each little spot here, we have a transcriptome, which is all of the gene expression values of all the genes in the genome. So it's one long vector of genes and values of whether they're up or down. And underneath that spot is a little image patch. And so we take that, we take that image patch and we encode it here, and we take the transcriptome and we encode it here. And then we use a contrastive loss, which basically forces these things to be aligned. And then once it's finished learning, we can take an image and we can convert the image to the latent space, convert the image latent space to the transcriptome latent space, and then go back out to the gene expression space through going backwards. And then we can predict the expression based on the image. And so this is how well we do at least for some cherry-picked best cases. Here's the original data, which shows a particular gene, the CYP3A4 gene, which is a gene in the liver that processes drugs and for drug metabolism and other kind of small molecules that you eat. So it's an important liver enzyme. And this is its spatial pattern. And this BLEEP method that I mentioned that we developed is able to predict pretty close to the original spatial pattern and get a 74% correlation. In general, we don't get a very high correlation, like let's say 20% correlation across all genes, but there definitely are some interesting examples of genes where we get very good correlation. And so again, this is not a solved problem. We can't perfectly predict the transcriptome from the image, but we definitely see some interesting correlation, and we're further working on this to improve it. Okay, so I could stop here if there's questions. And if there's no questions, there's one very short other set of slides that I can finish with. I'll just quickly mention a kind of another way that we're, as people in the field are approaching trying to understand these systems, and this is through simulation. So there's a project called the Virtual Human Development Project, led by these people. Maria's in my group, and Nozomu Yachi and Nika Shakiba are at UBC, actually in the bioengineering department there. The idea of this project is that we can create mathematical models that can grow cellular systems in silico, and then you can play with the parameters and see how things change. So for instance, this model can create one model that we've created can create like a 3D set of cells. Their model is bubbles, and the math is just bubble geometry. But the key thing is that there's an idea that you can do simulations of systems in the body, and if we improve that, we can kind of merge that into generally understanding the body. Okay, so that's it. I mentioned people along the way who have worked on the projects that I just described today, but lots of people in my group, and get funding from a lot of different places that supports our work. So I acknowledge that all of those people and funders as well.